Hello everyone, welcome to our channel Medical Mentor. I'm here today with a very fascinating topic in the field of medicine, that is immune system. As the topic of immune system is pretty vast, this video is going to be about the introduction to the immune system. I'm going to divide this topic into two videos, so let's start. What is immunity? In simple terms, immunity is the human body's remarkable ability to resist or withstand almost any organism or toxin that could harm its tissues and organs. Before proceeding forward, there are few terminologies that we need to be familiar with. What is antigen and what is immunogen? You may think these words have a similar meaning, but there is a subtle difference. An antigen is any substance that can bind to the products of an immune response, such as antibodies or T cell receptors. Essentially, it's a molecule that the immune system can recognize. However, the point to remember is that not all antigens that bind to antibodies or T cell receptors can trigger an immune response. But an immunogen is a substance that can provoke an immune response. Therefore, we can say that all immunogens are antigens, but not all antigens are immunogens, as antigens may or may not show immune response, but for an antigen to be called immunogen, it should provoke an immune response. The term antibody and immunoglobulin are used interchangeably. Antibody or immunoglobulin is a protein produced as a result of immune response against antigen. Epitope is the site of an antibody recognized by the immune system, and the part of an antibody that binds to the epitope is a paratope. Plasma cells are the terminally differentiated B cells that produce antibodies. MHC or major histocompatibility complex molecules are cell surface proteins that display peptide fragments which may be derived from pathogens or self proteins for recognition by T cells, ultimately playing a crucial role in the adaptive immune system. T cells can be classified as CD4 positive T cells, CD8 positive T cells, T regulatory cells, and memory T cells. So MHC class 1 molecule is present in all nucleated cells, and it presents peptide fragments to CD8 positive T cells, also known as cytotoxic T cells. Whereas MHC class 2 molecule is present in antigen presenting cells and presents its peptide fragment to CD4 positive T cells, also known as helper T cells. A point to be noted in this is that, as we know, MHC class 1 molecule is present in all nucleated cells, so antigen presenting cells, which are of course nucleated, have got both MHC class 1 molecule as well as MHC class 2 molecule. Antigen presenting cells are the immune cells that process and present antibodies to the T cells through MHC molecules, triggering adaptive immune response. Dendritic cells, macrophages, and B cells are the antigen presenting cells in our body. Opsonins are the immune proteins, such as antibodies and complement protein C3b, that coat pathogens, making them easier for phagocytic cell to recognize and engulf for destruction, and the process is known as opsonization. Active immunity results from your body's own immune system producing antibodies in response to a pathogen or vaccine, while passive immunity involves receiving preformed antibodies from an external source, providing immediate but temporary protection. Immune system organs can be classified as primary and secondary. In primary organs such as bone marrow and thymus, immune cells are produced and or are pre-processed. B lymphocytes are produced as well as pre-processed in bone marrow, whereas T lymphocytes are produced in bone marrow, but later on mature or pre-processed in thymus. In secondary organs like lymph nodes, spleen, Peyer's patches, and tonsils, immune cells interact with antigen. Immunity can be divided into innate, which is present at birth, and acquired or adaptive immunity, which is acquired throughout life. Acquired immunity is called natural if developed naturally after exposure to pathogen or antigen, whereas it is called artificial if immunity is induced through deliberate actions such as vaccination. Both natural and artificial acquired immunity can be divided into active if the body produces its own antibodies in response to an antigen, and passive if the body receives pre-made antibodies from another source. Example for natural active immunity would be a person developing antibodies against chickenpox after being infected by the virus varicella zoster. Example for natural passive immunity would be a baby receiving preformed antibodies from the mother through breast milk. Example for artificial active immunity would be a person developing immunity against measles and rubella after being vaccinated with MR vaccine. And an example for artificial passive immunity would be getting an injection of antibodies against rabies virus after being bitten by animal. This figure illustrates that the immunity that fights first against the pathogen or antigen is innate immunity, and the adaptive or acquired immunity gets activated only after a day or two. The principal cells of innate immunity are epithelial barriers, phagocytes such as neutrophil and macrophages, dendritic cells, complement proteins, and natural killer cells. The principal cell of adaptive immunity is lymphocytes, B and T. B lymphocytes are the ones that are terminally differentiated into plasma cells and they secrete antibodies. However, some of the innate immune cells like dendritic cells and macrophages help in activating and transforming T lymphocytes into effector cells, which is required for T cell function. So firstly, let's learn in brief about innate immune cells. Innate immunity is a rapid and non-specific defense. Though it is the first to defend our body against any antigen, it does not form any memory immune cells for that particular antigen. If the same antigen re-encounters us in future, it responds as it would against any other antigen. Therefore, there is no specific response for a particular antigen. Epithelial barriers such as skin, respiratory, and gastrointestinal tracts block the entry of microbes. I'm going to talk about the other innate immune cells in the next few slides. Innate immune cells such as neutrophils and macrophages are phagocytic cells. These cells can sense the presence of microbes as well as other offending agents, engulf them and destroy them. This process is known as phagocytosis. We can talk about phagocytosis in detail in other lectures, but here I just want to explain the process in brief. Three steps are involved. The first step is recognition and attachment, where microbes bind to phagocyte receptors. The second step is engulfment, where there is extension of cytoplasm around the microbes bound phagocyte receptors, and plasma membrane gets pinched off to form a phagosome, which contains the ingested particle or microbe. There is another membrane bound organelle present there called lysosome, which contains various hydrolytic enzymes. Now there is fusion of phagosomes and with lysosomes forming phagolysosomes. There is also production of reactive oxygen nitrogen species within these phagolysosomes. The third step is killing and degradation, where microbes are destroyed by lysosomal or hydrolytic enzymes and by generation of reactive oxygen as well as nitrogen species. Dendritic cells are found beneath the skin, lymph nodes, and many other tissues. We are already familiar with the fact that dendritic cells are one of the antigen presenting cells and help in adaptive immune response. But in addition to that, these cells have a rich collection of receptors that sense microbes and cell damage. After detecting danger, they stimulate the secretion of different cytokines, mediators that help in inflammation and antiviral defense. In a way, we can say that dendritic cells initiate innate immune response by detecting danger signals. But in contrast to macrophages, they do not destroy the agents on their own. Natural killer cells are one of the types of lymphocytes, and they play a role in innate immunity, so also referred to as innate lymphoid cells. Their function is to recognize severely stressed cells that may be virus-infected cells or tumor cells and kill those cells. They can also recognize antibody-coated target cells and lyse them. This is known as antibody-dependent cellular cytotoxicity. The killing mechanism is regulated by signals from activating and inhibitory receptors. As we can observe in the figure, a normal or healthy cell expresses a self-MHC class 1 molecule, which is recognized by the inhibitory receptors present in the natural killer cells. Thus, NK cells are not activated and there is no cell killing as it should be. The cell is normal. But when the cell is, for example, infected by a virus, there is exhibition of the viral peptide by MHC class 1 molecule, which gets engaged with the activating receptor of natural killer cells. NK cells get activated and there is killing of the infected cells. NK cells even secrete cytokines such as interferon gamma, which activates the macrophages and enhances their ability to destroy microbes. Talking in detail about activation of complement protein is a lecture in its own. So here we're going to discuss about different complement proteins which take part in innate immune response. Complement proteins are the plasma proteins which are present in inactive form. These proteins are sequentially activated by three different pathways: classical, alternative, and lectin. Alternative and lectin pathway of complement activation is a part of innate immune response, whereas classical is a part of adaptive immune response. C5a and C3a are responsible for recruiting leukocytes and cause inflammation. C3b acts as an opsonin which tags the microbe and helps macrophage to phagocytose it. After the sequential activation of different complement proteins through any pathway, there is formation of a membrane attack complex, which lies the microbes. So the innate immune cells basically perform two most important cellular reactions. First is inflammation and second is antiviral defense. In the inflammation process, phagocytic leukocytes such as neutrophils and monocytes or macrophages are recruited and activated to kill microbes. The antiviral defense is mediated by dendritic cells and natural killer cells. In the next part, we're going to discuss about adaptive immune system. Thank you for watching. This is the beginning of our wonderful journey ahead with you all. Don't forget to subscribe to our YouTube channel and hit the notification bell for more such educational content.